everyone, this is Ramalinga Prasad Kuppa. Welcome to my channel, Pharma World. Today's topic is Technology Transfer Part 2 from Sending Unit SU to the Receiving Unit RU. In today's topic of Part 2, we will discuss on the detailed responsibilities of the key players in the technology transfer. The sending unit is the originator of the transfer, and the receiving unit is the one where the transfer data is translated into commercial scale. SU and RU are the main participants in technology transfer. Let us see the characteristics of the key players. Transfer of technology is defined as a logical procedure that controls the transfer of any process together with its documentation and professional expertise between development and manufacture. As told above, the key players are the sending unit and the receiving unit. The WHO Technical Report Series number 961 dated 2011 Annexure 7 Guideline on Technology Transfer defines the transfer as a logical procedure with necessary and comprehensive documentation between these SU and RU for effective execution of transfer of data and scale up. Logical means that the transfer of technology should be sequential with a scientific logic for each step of the transfer. Two important participants in technology transfer are sending unit and receiving unit. Transfer is between the originator and the receiving unit. There could be more than two receiving units depending upon the number of sites of transfer. The capabilities of the SU and at the RU should be similar but not necessarily identical. Obviously, the capabilities of SU and RU should be similar so that the understanding is on the same platform. The capabilities can have similar characteristics but not necessarily exactly same. Facilities and equipment should operate according to similar operating principles. Infrastructure and operations also should be with similar principles and not necessarily the exactly same. Let us see the path forward. Detailed project management plan should be in place which identifies and controls all the activities identified at the start of the undertaking. This document is very important. The project management plan should have technology transfer protocol. The details of technology transfer protocol were discussed in part 1 of the technology transfer training module. As a separate part of file, the technology transfer, all necessary validation documentation should be provided to the receiving unit. Detailed risk assessment reports also should be provided. The SU and RU should jointly verify the qualifications of the equipment and impart necessary training. The sending unit, that means the originator, and the receiving unit, that is within the manufacturing section or any other third party also. The qualification of process and analytical equipment, necessary training should be done at this stage. The depth of training depends on the criticality of operation. Necessary training on safety precautions to be taken while operation should also be included. It is important to constitute a team from both SU and RU for execution of translation of data. The responsibilities should be clearly defined. There could be overlapping responsibilities too. In such cases also there should be clarity on individual responsibilities of SU and RU. As per mutually agreed timelines, the transfer of technology should start. Now, the transfer of data onto the shop floor should start. More elaborate data will lead to an effective transfer of data. Trial batches should be planned and executed. The data generated should be evaluated statistically for process capability. Minimum of 3 consecutive batches should be considered for trials. Statistically, you need this number to establish CP and CPK values. Let us see how QC and method transfer is done. Transfer of analytical methods should be part of the technology transfer. The sending unit should provide all QC related data in much detail. All raw material, packaging material, in-process material, intermediate material, final product specifications should be provided. Approved specifications for this list should be provided. Detailed test procedures for these items also should be provided. Non-compendial test methods should be transferred along with the analytical method validation reports. This point is very important. If the compendial methods are used, it may not be necessary to carry out the method validations. It does not mean that validations are not necessary for this. The analytical method gets listed in the compendia only after submission of complete validation data, which will be reviewed by team of experts for compliance. However, it is necessary for verification at RU site for suitability to their laboratory conditions. You need support of USP 1226 chapter for this. Necessary impurity standards and finished product standards should be provided by SU. If it is not available in the open market, it is the responsibility of the SU to provide the impurity standards and finished product standards to RU. The certificate of analysis and its validity should be mentioned on the certificate of analysis. Validated analytical methods for establishing the residues of wash water also should be provided by SU. The RU should review all these methods and confirm suitability for routine use. Let us see how an experimental design works. Let us evaluate this table. For QC technology transfer, let us see how the design of transfer works. In this typical example, provided in the guideline, while transferring the technology for identity test, sample preparation and data interpretation should be focused. One determination is adequate. No other evaluation is necessary. Assay test has to be specific to the product estimation. 18 test results are required for replicate data for transfer. Using different instrument, different brand columns, separate sample preparations should be considered. Direct acceptance criteria may be adequate with the mean value of the results and variability. Statistical evaluation should cover the t-test with a specification of not more than 2% and 95% confidence. The numerical limits in the statistical evaluation are arbitrary and can be revised as justified. You may have to try a couple of examples to get into the t-test and 95% confidence procedures. Cleaning verification also takes important place in technology transfer. requirements are tabulated in this table. Swab material plays very important role in recoveries of residues. It is recommended to use the same type of swab at both SU and RU. Spiked samples levels should be within plus or minus 10%, whichever is greater. Direct acceptance criteria is adequate. No further statistical evaluation is necessary in this case. Yet another example for impurity, degradation pathways and residual solvents is provided in this table. Response factors, LOD, precision and accuracy for spiking experiments should be considered. Data from 12 points from 2 analysts, 3 lots in duplicate is a requirement. Different days, different instruments and columns should be considered in setup at RU. Direct and statistically derived requirements should be complied as prescribed here. All three tables above are few examples. Depending upon the criticality of the method transfer, more elaborate evaluation may be made. But this model works very effectively. Let us see how the premises and equipment are handled during technology transfer. The premises should be suitable for technology transfer. SU and RU team should evaluate suitability through a risk assessment report. The risk assessment should include inherent risks of the manufacturing process, example reactive chemical hazards, exposure limits, fire and fire explosion risks, health and safety requirements to minimize operator exposure, example atmospheric contamination of pharmaceutical dust, emergency planning considerations, example in case of gas or dust release, spillage, fire and firewater runoff, waste streams disposal and recycle. Both the sending unit and the receiving unit should discuss the suitability of all equipment for technology transfer at receiving unit. The risk assessment should discuss all these items in much detail. The RU should perform a gap analysis to identify requirements for adaptation of existing equipment or acquisition of new equipment or a change in the process to enable the RU to reproduce the process being transferred. Data submitted by the sending unit should be reviewed by the receiving unit for suitability and take necessary correction after verifying out the gap analysis. This supports to enable the RU to reproduce the process on routine basis. GMP requirements should be satisfied and intended production volumes and batches, example same scaled up or campaign, should be considered. The GMP requirements have to be complied with all through. Let us see what kind of documentation is required for technology transfer. Entire technology transfer is established by documented evidence of various documents from several stages of execution of transfer. Documentation is the objective evidence of technology transfer. Complete documentation for all stages from start to finish should be available. The formal documentation system should consist of key tasks, documentation provided by the sending unit, and the transfer document. Guideline recommends to use these aspects in a tabulated form with these headers. This should track the requirements in a sequence so that there are no missing points. All the documents should be submitted in sequence for easy reference. You will see some typical examples in coming up slides. Let us see the example documents. The key task, the project definition should be clear. The documentation provided by the sending unit should have a project plan and quality plan where separate documents are required. Necessary protocols, risk assessment reports, gap analysis should be submitted by the sending unit. The project implementation plan, the TOT, that is technology of transfer protocol, should be the transfer document. The quality agreements on plans and layout of facility, buildings, qualification status like DQ, IQ, and OQ, and the reports should be provided by the sending unit. Side by side comparison with receiving unit facility and buildings, gap analysis, qualification protocol, and reports should be done. Health and safety assessment should be done. It is a product specific waste management plans, contingency plans also should be provided by the sending unit. Skill set analysis and training, all necessary SOPs and training documentation should be provided, which is product specific, product operation specific, analysis and testing, training protocols and assessment results should be the transfer documentation. Analytical method transfers should be transferred with all specifications and validation reports, including in-process quality control test methods and specifications. The analytical method transfer protocol and report should be the transfer documentation. The key task of starting material evaluation, all the necessary specifications and additional information on the vendors, if available, should be supplied by the sending unit. Equipment selection and transfer, inventory list of all equipments and systems, including makes, models, qualification status like IQ, OQ, and PQ, the drawings, manuals, logs, SOPs, etc. All those things should be the part of the documentation provided by the sending unit. At the same time, the comparison with the equipment in RU in terms of its makes, models, qualification status should be done. The gap analysis, qualification and validation protocols and reports should be checked in the transfer documentation. The cleaning validation, including solubility information, therapeutic doses, category like toxicology, existing cleaning SOPs, validation reports, chemical and micro agents used, recovery studies, etc. should be the part of the documentation provided by the sending unit. Product and site specific cleaning SOPs at RU, cleaning validation protocol and reports should be checked in the transfer documentation at RU. Under key task, process transfer, manufacturing and packaging, the documentation provided by the sending unit should have reference batches, development reports, manufacturing process rationale, history of critical analytical data, rationale for specifications, critical manufacturing process parameters, product stability data, current master batch manufacturing and packaging record. The transfer documentation should contain the history of process development at RU. Experiences at RU should be recorded for future reference. Provisional batch manufacturing document and provisional batch packaging record. These two have to be developed by the receiving unit. Description of process at receiving unit, process validation protocol, and reports. I hope that the information on intricate requirements of technology transfer is understood well. Since this is a short video, the intent is explained in brief. For more details, the WHO Technical Series Report number 961 dated 2011 Annex 7 may be studied. All these intricate points should be incorporated in the technology transfer policy. Effective procedure would lead to a good technology transfer. Here, the sending unit could be the development team of the originating unit. Thanks for watching. For more videos, please do subscribe, like, and share. Thank you.